Hi, I'm Christy Morrison-Thomas, and I work with the CarbonTIME project at Michigan State University. This infomercial focuses on CarbonTIME, transformations in matter and energy. First, I'll share an introduction to the CarbonTIME project. Then, I'll share our research base, followed by our instructional design principles. I'll use the CarbonTIME plants unit to walk you through an example, and then connect CarbonTIME to its most common use in high school biology. CarbonTIME's goal is to support students in developing into scientifically literate citizens who can apply scientific knowledge and practices to socio-scientific issues, including use of environmental resources, global climate change, and nutrition and health. Our goals include helping students be prepared for future learning, recognizing that what we understand about the world continues to change. CarbonTIME is an NSF-funded project with research and development work extending over more than the last 10 years. The first of 2 5-year grants supported research into students' understanding of carbon-transforming processes and the initial development of teaching units. The second 5-year grant supported using these units through research practice partnerships that designed and implemented sustainable systems at scale for shifting teacher practice to support students' three-dimensional performances. Our principal investigator is Dr. Andy Anderson, now Professor Emeritus at Michigan State University, and our program is now living with other high-quality, research-based science curricula at MSU's Create for STEM Institute, and supported by NGPBL's professional learning cadre. The 3 legs of the stool is the fundamental design of our CarbonTIME project. We believe that 3-dimensional teaching requires systemic supports through curriculum and aligned multidimensional assessments, as well as coordinated, parallel professional development. We think of this professional development as helping teachers view CarbonTIME units as a toolkit, not a script, that will help their students master rigorous 3-dimensional performances. Together, all of this proceeds through networks which provide collegial support, encouraging changes in student learning and teacher practice. There are 6 CarbonTIME units designed to support students in tracing matter and energy through living and earth systems. Units include 4 units on carbon-transforming processes at the macroscopic scale, including systems and scale, animals, plants, and decomposers, as well as 2 units on carbon-transforming processes at the large scale, including ecosystems and human energy systems. All 6 units are finished and fully available for free on our website. A login is required for accessing password-protected assessments and teacher answer keys. The most common classroom use of CarbonTIME units is as part of a 1-year high school biology course. These units comprise 1 semester's worth of instruction. Next, we'll take a look at the CarbonTIME research base. Learning progression research is an important part of why CarbonTIME works. This learning progression research has helped us understand how students develop 3-dimensional knowledge and practice. Our research has supported the defining of 3 different learning progression frameworks. 1 around how students construct explanations for carbon-transforming processes, 1 around inquiry practices related to designing investigations and interpreting data, and a 3rd framework around how students make sense of large-scale systems that require interpretation and predictions around data sets and models. These 3 learning progressions have informed the design of the CarbonTIME instructional model and all of our student and teacher-facing materials, so that we're intentionally supporting students' development of more sophisticated understanding, practices, and principle-based reasoning. Further, the learning progression research is part of our teacher professional development. We're supporting teachers in developing a learning progression stance, meaning that they can use tracing matter and energy as helpful ways of interpreting student ideas across differing contexts, from burning flames to growing children. Our learning progression research helps us understand students' initial sense-making. We call these Level 2 and Level 3 accounts, and almost all high school students entering biology make sense of living systems in these ways. Students describe plants and animals as actors whose actions are enabled by food and oxygen, rather than as systems that transform matter and energy. For example, they think about nutrients and energy cycling in ecosystems. Plants use sunlight to make food. Animals eat plants and other animals as they participate in food chains. And then something called decay returns nutrients and energy to plants to start again. Students also think about oxygen-carbon dioxide cycles, in which plants change carbon dioxide into oxygen through photosynthesis, because animals need it. And then animals change oxygen into carbon dioxide because plants need it. This kind of story, in which one invisible gas in the air changes into another, arguably makes more sense than thinking about an invisible gas changing into wood in a tree. Part of our job is to help students feel dissatisfied with their initial, sensible stories and help them build deeper understanding based on canonically correct scientific models. This means supporting students in moving to Level 4, or NGSS Goal Understanding. This reflects a single carbon cycle. Plants, animals, and decomposers are all subsystems that transform matter as it cycles and energy as it flows through ecosystems. We want students to use these coherent scientific accounts, including principles such as conservation of matter and energy at multiple scales. Then they can explain that trees use inorganic carbon dioxide from the air along with water to make organic sugar molecules that are used to build the tree's mass. At the end of CarbonTIME units, students use these models, including organic pools and fluxes, to explain natural phenomena such as growth of organisms and changes in ecosystems. This graph provides evidence that CarbonTIME works. It shows us student results on CarbonTIME post-tests, which have three-dimensional assessment items similar to those in the New Science MSTEP. The blue bars show post-test results for classrooms not using CarbonTIME units and professional development. In these classrooms, after a year of biology, students have a Level 2 understanding. The pink bars show results for classrooms that were using CarbonTIME. After a year of biology, most students have a Level 3 and Level 4 understanding. So we can see that there is a much higher achievement for the CarbonTIME classrooms. More students are reaching sophisticated science understanding and practices. Our quantitative analysis team statistically analyzed tens of thousands of student assessments to identify 3 variables that statistically explain differences in students' learning gains. As you might expect, lower school resources, as measured by higher % free and reduced lunch, are associated with lower learning gains, but that's less than 4% of the explained variance. Students' prior knowledge and experiences, as measured by pre-test scores, explains about 10% of the differences in student post-tests. We also found that lower pre-test scores are associated with higher learning gains. In other words, CarbonTIME lessens achievement gaps. Finally, individual teachers are by far the largest factor affecting student learning gains. The teacher statistically explains a third of the variation among students' learning gains. This means, of course, teachers and their professional learning experiences matter. By studying classroom videos, we have found that in higher student learning gains classrooms, the teacher is explicit about the purpose, always connecting activities and dialogue to the unit driving question. Evidence from investigations is the authority, not just what the teacher says. Students have agency, meaning that they are figuring out the unit phenomena, they are building knowledge. There is precision in language, meaning that the teacher uses language to connect macroscopic observations with atomic molecular scale explanations, and teachers and students clarify when others conflate matter and energy. And there's a bridge between students' initial ideas and scientific accounts, building students' environmental literacy in ways that supports their identity development as science people. Through interviews with these same teachers, we also know that they engage in sustained sense-making around CarbonTIME that is student-centered. They perceive alignment between CarbonTIME goals and goals in local contexts, which is helpful because they're not trying to do CarbonTIME and also do other things on the side to prepare kids for required one-dimensional fact-based exams. And they believe students can be motivated to master three-dimensional learning goals, because figuring out natural phenomena and mastering three-dimensional performances is hard work. Successful teachers believe students can and will do this work. In our research program, teachers participated in a 2-year professional development course of study with a network of colleagues, as mentioned on the 3 legs of the stool slide. Initial findings suggest that district-based networks, like the one in Seattle, were most successful in shifting teacher practice, and teachers in those networks experienced greater alignment among local expectations and CarbonTIME goals. In other words, networks and the local context matter. CarbonTIME has a partnership with the MEA to help develop this kind of district-level partnership here in Michigan. Next, we'll look at design principles, an instructional model that combines inquiry and apprenticeship. As you know, the next generation science standards represent three-dimensional science engagement. This framework asks for multidimensional performances from students. Science students blend together what scientists do, the science and engineering practices, with what scientists know, the disciplinary core ideas, with how scientists think, the cross-cutting concepts, in an effort to figure out natural phenomena together. This means that in CarbonTIME classrooms, instead of learning about combustion, students figure out what happens when ethanol burns. And instead of learning about photosynthesis, cellular respiration, or biosynthesis in plants, students figure out how radish seeds grow and develop new body parts and move and function. We think about the NGSS 3 dimensions in CarbonTIME using this image of the triangle, which is the core of our instructional model. So, as students are figuring out natural phenomena, they are distilling many observations into a few important patterns. These are the inquiry practices. They're designing investigations, collecting data, analyzing that data together, and making evidence-based arguments. Students then use key models to construct explanations for the phenomena, applying their model to investigation and unit phenomena, and eventually to new sets of phenomena that they have not encountered in the classroom. This approach is the core of our unit design, our instructional model, which is consistent across all of our units. It shows how students figure out unit phenomena. Observations, patterns, and models are tied together by investigations and arguments from evidence, as well as model-based storylines that trace matter and energy through systems. The color coding here supports students and teachers in where they are in a unit, and we'll look at this more shortly with our example in the plants unit. Another design piece across units is what we call the 3 questions, through which CarbonTIME scaffolds important cross-cutting concepts of scale and matter and energy. In order to explain a carbon transforming process, students must connect the macroscopic with the atomic molecular scale. To explain where molecules are moving to and from, or the matter movement question, how atoms and molecules are being rearranged, we call it the matter change question, and what is happening to energy, the energy change question. You'll see how these 3 questions are scaffolds as we look at the plants unit next. Finally, our units are intentionally designed to promote discourse in ways that foster student sense-making and 3-dimensional performances. Our research findings suggest that there are key features of classroom discourse in high learning gains classrooms. So we focus in PD and in the educative features of our curriculum on this discourse routine. The discourse routine is an intentional sequence of private and public talk and writing surrounding each process tool, in which we start with many different ideas and questions, divergent thinking, and come to consensus or convergence around some ideas and questions at different points in the unit. There's an introduction in which the teacher and students connect to the purpose of figuring out the driving question. The students then have private writing and thinking time to identify their own ideas and questions. Then students share ideas in small groups or with partners. Then the whole class shares ideas together, followed by a consensus-seeking discussion accompanied by public writing, in which some convergence is reached. For example, agreeing on some unanswered questions that remain after the investigation is completed, during an evidence-based arguments lesson. Next, we'll look at CarbonTIME Plants unit as an example. As we quickly overview one of our units, I'll share this slide to help orient us to the learning goals of the unit. Just to be clear, we do not provide this slide to students at the start of the unit, but to help you see where we're headed. In the plants unit, students figure out that plants use the food they make in two ways. Glucose produced in the leaves during photosynthesis travels to all of the cells in the plant, and cells use that food for growth through the process of biosynthesis, and for energy to complete living functions through the process of cellular respiration. There are several teacher and student-facing tools that coordinate with our instructional model and help keep track of progress over the unit. This teacher-facing unit storyline chart helps teachers see the whole unit in just one page. The other example is the teacher's answer key for the plant unit's learning tracking tool, which is designed for individual students and the class as a whole to keep track of what they've done and figured out through the unit. You may be familiar with it as a summary chart or a story so far chart. This is the instructional model in the CarbonTIME Plants unit. We'll start with a quick snapshot of Lesson 1. In CarbonTIME, most units are comprised of 6 lessons, each of which is made up of 2 to 6 activities. I'll be providing an overview of key activities in which students engage with particular process tools. These are the activities in which students and teachers complete CarbonTIME Discourse Routines. The unit begins with students observing a time-lapse video of plants growing, prompting them to consider this familiar and important natural phenomena. Plants get bigger, they get new body parts, they move. The Expressing Ideas tool provides support for students to draw on experiences and ideas that they have and to ask questions that will help guide the next steps in the lesson. Specifically, it begins prompting students to consider how they can make sense of this natural phenomenon by thinking about matter, what goes in and what comes out. These ideas and questions are the source of the unit's driving question board. Each unit also begins with a student storyline reading, which highlights a scientist in the field. In the plants reading, we read about Asima Chatterjee, a biochemist who worked in India in the mid-1900s. The purpose of these readings centers around developing students' science identities. It helps them see how actual scientists in the field took on particular roles as questioners, investigators, and explainers. The readings connect scientists' work to work students engage in during a CarbonTIME unit. For example, in this first lesson, where they're asking many questions that shape their path throughout the unit. Later lessons, in which they act primarily as investigators. And finally, as they put their understanding together to build model-based explanations at the end of the unit. Returning to our unit instructional model and our brief overview of the plants unit, we'll move forward to Lesson 3, Investigating Plants. In Activity 3.1, students use a predictions and planning tool to design investigations that will help them figure out how their plants are growing and moving. They start by returning to their initial ideas about how matter moves and changes, and how energy changes when plants grow and move. Notice that those are the CarbonTIME 3 questions. Of course, this intentional return to and revision of ideas is an important part in developing deep understanding. Students are prompted to consider how key tools, including digital balances and bromothymol blue, which changes color in the presence of carbon dioxide, can help them detect changes in matter and energy. They use this thinking to design investigations. They'll explore what happens when plants and BTB are in a closed container overnight, in the light and the dark. And they'll explore growing, drying, and massing radish plants. Through their investigations, students make these observations and identify these key patterns across class data. When plants grow, they gain more dry mass than the soil loses. Blue BTB turns yellow when plants are in the dark, or plants emit carbon dioxide in the dark. And yellow BTB turns blue when plants are in the light, meaning that plants absorb carbon dioxide in the light. Then they use these patterns as evidence on which to base certain claims. For example, they use evidence that the mass of the plant increased while the soil, either gel or paper towel, mass remained unchanged. To conclude that atoms from outside of the plant moved into the plant, increasing its mass, and that atoms must not have come from the gel or paper towel. They also identify key unanswered questions. For example, they wonder, well, where did the atoms that moved into the plant come from? And how did the atoms move into the plant? And what kind of atoms are moving anyway? These key questions at the atomic molecular scale position students to need to use a molecular model to construct an explanation. Here, you can see the way that the three questions scaffold students' thinking across activities, as this row helps students focus on the matter moving question, and other rows that we don't see here focus on the matter change question and the energy change question. Next, in lessons 4 and 5, students explore molecular models and use them to explain all three processes required to fully explain what happens when plants grow, move, and function: cellular respiration, photosynthesis, and biosynthesis. Students use molecular modeling kits to concretely visualize carbon-transforming processes like photosynthesis, starting with the carbon dioxide, water, and light they know the plant is taking in, and using the kits to solve their unanswered questions from the evidence-based arguments tool. They see that atoms last forever and energy lasts forever. Students then build an explanation for how does a potato plant make the food it needs to grow and function using the explanations tool. The front side of the explanations tool is a graphic organizer designed around the three questions. Coordinating PowerPoint slides support thinking across scales, including animations in which students can predict and then see molecules moving in the plant. Students engage in molecular modeling and constructing model-based explanations for each carbon-transforming process: cellular respiration, photosynthesis, and biosynthesis. Repetition is a feature, not a bug, of the unit design. Our learning progression research and classroom-based research indicate that students need repeated, scaffolded practice to move from level 2 and 3 accounts, which focus on plants as actors or which conflate matter and energy, for example, sunlight is changed into food, to a sophisticated account that traces matter and energy across scales to explain phenomena. Then, in Lesson 6, students work to apply their model to new examples and to put together complete explanations about all plants. For example, by this point in the unit, most students are beginning to wonder, well, what about cacti? What about plants that grow in the water? What about pine trees that have needles? In Lesson 6, students explore these questions using readings and explanations tools that don't have the graphic organizer on the front and are without explicit framing around the three questions. In this example, they explore the prickly pear. Other examples are Spartina and the lodgepole pine. At the end of the unit, the CarbonTIME assessments include three-dimensional item clusters, similar to the New Science MSTEP, that focus on inquiry practices as well as explanations practices. Now let's look at the role of CarbonTIME in a high school biology course. The most common implementation of CarbonTIME units is as one semester of high school biology, when Earth and space science standards on climate change are incorporated into biology along with the life science standards. The 6 units take around 3 weeks each, although animals, plants, and decomposers have some identical activities. Teachers are prompted to skip these or to use them in modified ways for review. Then, second semester biology focuses on tracing information, covering genetics and evolution topics. This slide represents Novi's Semester 2. A team of CarbonTIME teachers in Novi, East Kentwood, Midland, and Seattle have worked together to extend CarbonTIME scaffolds through Semester 2. They have developed genetics units centered around Axolotl, which is a fully aquatic salamander, as well as on human albinism. Then, they rely on the NGS storylines unit, why don't antibiotics work the way they used to, for evolution standards. This team of teachers has extended CarbonTIME scaffolds so that learners experience a coherent, year-long biology course. Here, we can see an Expressing Ideas tool at the beginning of the genetics unit, asking students how and why they think axolotls are different. Teachers and students also rely on a set of 3 questions for genetics, which, like the CarbonTIME 3 questions, help scaffold students in tracing information across scales, from macroscopic observation of traits at the organismal level, through cells, to the biological molecules and mechanisms inside of cells. Thank you for your listening, and thank you to our funders.